Oops. New screen recording. Oh, it is recording. Okay, so we were right here. We were talking about the basic functionality of epithelial tissues, right? And then we'll talk about the locations of epithelial tissues, and then we'll start getting into the specific tissues. I'm going to go ahead and tell you that there are a couple of resources. I don't know if you looked at them yet. I mentioned them to you last time. If you go to the links page of melanieb.com under Histology, there's a link to a really, this is the college that I taught at before here. This is another site that has some good histology practice activities in which you can do identification. And I told you guys that there's like some pathology slides. You don't have to, I'm never going to test you off with pathology slides, but it is kind of interesting to see the difference between, for instance, normal brain tissue and cancerous brain tissue. He also has some good normal slides on there as well. But the one that I'm going to be using today, and you might want to use in the future, is this one. You can click on epithelium, and the thing that I like about it is that it has both labeled and unlabeled so that you can make flashcards out of them. So you could fold them over and make flashcards out of them if you wanted to. So I'll be like using this in addition to the textbook photos today as we go through. Good? Okay, so where do you find epithelial tissue? Well, we already kind of talked about it. You find it basically at the boundaries or surfaces of things, not just external surfaces, but also internal surfaces. So with that in mind, does it surprise you that the superficial surface of a serous membrane will be an epithelium? Does that surprise you? And the tissue that is lining the heart should also be an epithelium. The stuff that's on the surface of my eyeball, up the urethra, even if I could get my finger small enough, sticking it down a pore. That's all epithelial tissue, right? So lining body surfaces, covering body surfaces and lining body cavities. And then there's this weird epithelium that isn't necessarily near the surface. And don't worry, I won't make you memorize the slides. We'll just talk about it conceptually. All glands are also made of epithelium, even if they're not at the surface. Like, for instance, the pituitary gland deep below my brain, my thyroid gland, pancreatic tissue, that's all epithelium as well. Usually at the undergraduate level, you don't have to learn what those actually look like because they just look like a mess, honestly. So we'll do mostly covering and lining epithelium, the ones that are surfaces. Okay, so let's talk about the characteristics of an epithelium. It doesn't matter which epithelium you look at. Let's start with just the fact that they have what we call polarity. Epithelial tissues have one side that is different than the other side. So there is a free surface and a not free surface. And by not free, I mean that it is going to have something behind it. So do you see in this tissue that this has a free surface and then a not free surface that has what kind of tissue do you imagine is going to be underneath it? It's going to be a connective tissue. I'm not going to ask you to identify it in here, but conceptually that means that this tissue, unlike, for instance, this connective tissue, do you see this? This tissue has a top and a bottom, right? One side is different than the other. They call that characteristic polarity. So when you took chemistry and they talk about polar molecules, it means it's different on one side than the other, right? It's positive on one side and negative on the other. That's what this is implying as well. So I've got two sides of an epithelial tissue. I have the free surface, which is called the apical or apical surface. And the apical or apical surface, that implies top like an apex, but it really doesn't have to be on the top, just so you know. There's really no upside down for tissues in the body. So that's the apical or apical surface, and then this bottom surface right here is called the basal surface, as in base, bottom. Does that make sense? So the apical and basal surface. So now let's look at it on a real tissue. So if you look here, epithelial tissue, the easiest way to identify it is always look for the free surface. Look for the area where there is not tissue, and then find the tissue that is at the edge of it, right? So this is the apical surface, and this is the basal surface of that tissue, right? So, but we could do it with any of them. Apical surface, basal surface. Okay, now, underneath the basal surface, you have the stuff that sticks the epithelial tissue to the connective tissue, which is collectively called the basement membrane. Okay, so here's what the basement membrane is. If you go with a metaphor of tiling, whether you've ever tiled anything or if you watch like home improvement shows, what you're going to do is you're going to like deal with the subfloor, the superficial surface of the subfloor, and then you're going to put tile down on top of it. But before you do that, you're going to put a layer of adhesive on. and then you're going to put the tiles right next to one another. Does that make sense? So in this metaphor, the tiles, of course, are the cells of the epithelium, right? And the adhesive that you are dealing with is going to be the basement membrane. And the basement membrane is not made of cells. It's secreted by cells. It's proteins and sugars and sticky stuff. And think of it as like a two-part adhesive. Have you ever seen the two-part adhesives that it's not sticky unless you get both parts together? So what happens is the connective tissue secretes and the epithelial tissue secretes. And then when they come together, they produce that sticky surface that sticks the epithelial tissue to the connective tissue, okay? So it's acellular. Does that make sense? It's not composed of cells. You're not going to see any nuclei in there. And it's called the basement membrane. Here is what a basement membrane looks like if you can, I mean, sometimes it stains really well and sometimes it doesn't, but you can see it super well right here. Do you see that basement membrane? Even if it doesn't stain very well, it is there, right? Because there's a connective tissue and an epithelial tissue. So even though you can't see it super well here or here, when you get to the bottom of the epithelial tissue, there's a basement membrane underneath it. So here's something that students have trouble with. How about this terminology? When I am looking at a tissue and I point to this side and I say, name this surface, then the answer is the basal surface. But what if I say name this structure? That's the basement membrane. So it's my job to write a clear question, and it's your job to answer what I asked. Okay? So if you, for instance, looked at this slide, right? What is this surface? Basal surface. And what is the structure? Basement membrane. And now somebody use anatomical terms to describe for me the relationship of the basal surface and the basement membrane. Can anybody do it? Go ahead.
The basal surface is superficial.
Yes, the basal surface is superficial to the basement membrane, right? So they are separate structures, even though they're really close to one another. Go ahead.
But on the top, that's not true, right?
No, it is true. I mean, you're always going closer to a surface, right, when you're doing superficial and deep, right? I mean, it's kind of arbitrary what surface you're talking about, but we have to assume that you're talking about the free surface. Yeah? Does that make sense? Okay. So is that okay? So I will try to write that very clearly, but you just have to sort of listen to what I'm writing. And sometimes it's really easy to see, and sometimes it's not very easy to see, but it's always there. Okay, so that's this whole part that we just talked about, the polarity and the basement membrane, and we'll go back to these. So what else is always true about epithelial tissues, especially about surface epithelial tissues? The thing that's important about epithelial tissues, in addition to them having a free surface that's always easily identifiable, is that the cells are always right smack against one another. Like the tiles are right next to each other. Okay, that means that there's really no room for what we call extracellular matrix between them. And that's not true with a connective tissue. So let's look at the difference. So this is an epithelial tissue. Do you see it down there? Cells right next to one another. This is a connective tissue. There's a whole bunch of room for stuff between the cells, right? So an epithelial tissue has the cells right next to one another. So you can see it here, right? You can also see it here. The cells are smashed right up against one another. There's no room for matrix. This is also an epithelial tissue, a different type that we'll talk about in a second. And the cells are right next to one another. Okay, so there is really no room for much of anything between the cells. They're closely packed, little or no matrix, which brings us to another idea. The cells, epithelial tissue is usually what we call avascular. What does that term mean, even if you're not familiar with it? No blood vessels. So if I did a section of epithelial tissue, I'm not going to get a cross-section or a long section of a blood vessel in it. It does not have blood supply going directly through it, okay? Does that make sense? This connective tissue certainly does, but the epithelial tissue does not. So now, no matter which surface epithelium you look at, you're not going to get a cross-section or a long section of a blood vessel, which actually is kind of strange because I'm going to tell you something else about epithelial tissue, and the two things seem like they could not possibly both be true. The other thing about epithelial tissue is that it has a very fast rate of cell division and regeneration. It's a very active tissue. So doesn't it sound like those two things would have a hard time being true at the same time? No blood supply, but a very active tissue? So yeah, because of course oxygen and nutrients are brought by the bloodstream. So how the hell do you get around that? Well, the way that you get around it is simply because epithelial tissue, even the thickest epithelial tissue, is microscopically thin, so it's very, very thin. So even your skin is very, at least the living parts of your skin are very thin. And the tissue beneath it is very vascular. Okay, so how can I get nutrients in a very vascular tissue? Well, what happens is this tissue is richly supplied with nutrients and oxygen, right? The tissue on top of it is very, very thin. So it's like taking a sandwich. It's like, imagine you made yourself a grape jelly sandwich and you set it on the counter, but somebody left a puddle there on the counter. If you gave yourself a minute, wouldn't the top slice of bread be purple, right? And wouldn't the puddle also be purple if you gave yourself a minute? That's true as long as you don't have very far to go, but now do 12 grape jelly sandwiches and the top slice is not going to get wet, right? Okay, so as long as you're microscopically thin, then you can actually get your nutrients and oxygen via diffusion without a direct blood supply. Does that make sense? So it goes straight up and straight back down, straight up and straight back down, okay? That wouldn't work, for instance, for a chunk of cartilage, which is not microscopically thin. They're like big old chunks of cartilage in the body. So it is avascular, but it typically has a rapid cell division and regeneration, and that is good because if you put it at the surfaces of things, those are going to be the wear and tear areas. You're constantly rubbing cells off. Before I started teaching, I worked at a DNA fingerprinting lab, and if I had enough money, I could figure out that every single one of you was sitting in this chair because you always leave behind cells. You always do. You guys have seen Gattaca, right? Gotta be careful, like suck up those little eyelashes and everything. Okay, so you're always leaving stuff behind. You're always rubbing stuff off. And you need to regenerate relatively quickly. So on the surface of your skin, or like if you eat Captain Crunch for breakfast, you know you're doing some mouth damage, right? I don't know why the hell that cereal has to injure us all, but it's worth it, isn't it? It's totally worth it. Every single time you're like, oh, worth it. But by lunchtime, you can't feel it at all, right? Capable of rapid cell division and regeneration. So all of those are, the vagina is lined with epithelial tissue. It needs to regenerate because if you're doing right, there's friction involved, okay? The penis too, all of the external parts of the body are rapid cell division and regeneration. Now, that's both good and also potentially clinically risky because what do you think is more likely in tissues that are capable of rapid cell division and regeneration? Yes. Of the 4 tissues, this is the one that is the most likely to become cancerous because if you're constantly dividing, just statistically, you're more likely to lose control of cell division, and it really only has to happen once, right? Okay, so where do people get cancers? They get them in the skin, they get them in the lungs, but really it happens in the epithelium of the lungs. They get them in glandular tissue, like breast tissue, which is epithelium, like the thyroid, like the pituitary gland. All of those are cancers of epitheliums, or epithelial tissues, I should say. Does that make sense to you guys? Now, I'm not saying you don't get cancer anywhere else, because you can get cancer anywhere, honestly. But epithelial cancers are by far the most abundant of the 4 tissue types. Okay, now what else is true? Even though it's avascular, it is highly innervated. This word may not be familiar to you, but do you have any idea what that probably is implying? Innervated? Yeah, lots of nerves going to and from, right? Which is really good when it's at the boundaries of things because it's really good to be able to sense what is out there, right? So you have temperature receptors and touch receptors and pain receptors and all kinds of things. And okay, so we got closely packed, we got little or no matrix. The only thing, I'll come back to the hemidesmosomes. We'll talk about that. It's associated with the basement membrane. So now, just with your level of knowledge from lecture and lab, you should very easily be able to look at this and say, okay, that's definitely an epithelial tissue, right? And label, I mean, you don't have to do it now, but label the basal surface and the apical surface and the basement membrane. And you should even be able to tell me what kind of tissue this is below. It's connective tissue, exactly, right? So you don't have to do it now, but you can do all of that. Okay, so now let's move to how epithelial tissues are identified, labeled, and named. Luckily, epithelial tissues have a naming convention that they use most of the time. There's always an exception. And it's a 3-name naming convention. So there are going to be 3 words, and it's going to be, for instance, simple squamous epithelium. The first word refers to if I start from the apical surface and go to the basement membrane, how many layers of cells will I encounter? If it's only one, then it's called a simple tissue. So that's a simple tissue. This is a simple tissue, right? This is not a simple tissue, okay? This is a stratified tissue. Stratified means layered, okay? And it doesn't matter how many layers, anything more than one will be considered stratified. Is that okay? So the first word is simple stratified, or there's also one other thing, which is pseudo-stratified, which it looks like it's stratified, but it's really not. And I'll teach you that one individually when we actually get to that tissue, okay? So simple versus stratified or pseudo-stratified is the first word generally. And then the second word has to do with the shape of the cells at the apical surface of the tissue. So there may not be more than one layer, but if there is, you're always going to look at the apical surface. If the cell from the side looks like a fried egg squashed flat, that's a squamous cell. If the cell looks like a cube and usually has a nice round nucleus, that's a cuboidal cell. If the cell is taller than it is wide, and often, but not always, has an oval nucleus, then it's columnar. So now let's just look at these tissues, okay? So this one. Can you tell this is an epithelium? Yeah. Can you tell it's simple? What would you guess is the shape of the cells? Cuboidal. So you would call it simple, cuboidal, and the last word is always epithelium. Students very frequently leave off that word and they say simple, cuboidal, but you just gave me two adjectives and no noun, right? So what you need to tell me is it a simple, cuboidal kumquat, road sign? No, it's an epithelium, okay? So don't leave off the word epithelium because students do it all the time and they get dinged on it. So I need a noun, okay? So that's a simple, cuboidal epithelium. How about this one? Simple, yeah? Columnar, right? Okay, so you try to get to the point that you've looked at enough of them that you start becoming confident. That will not happen by just looking at the pictures in your textbook, which is why I'm trying to provide you with some other resources to look at. If you just look at this picture, then all you're doing is memorizing a photo. You're not actually learning what simple columnar epithelium looks like. Okay, so this one's simple columnar epithelium, and so now we will move on to looking at the individual types of simple tissues. Okay, so when we have simple versus stratified, oh, hold on, let me show you simple squamous. Here is a simple tissue. It's very hard to see that there's anything to look at because there's not much of it. Can you tell that this is an epithelium because there's a free surface? Yeah. Can you tell it's simple? Okay, and it's going to be squamous, right? The cells are squished flat. This is the hardest one to find because there isn't much of it when you're looking at it. Okay, so that's a simple squamous epithelium. Go ahead.
The cells on the bottom of it, though?
This is a simple cuboidal.
Okay, so
Yeah.
it's the same tissue, but one side.
No. This is a different tissue. This is two different epithelial tissues wrapping two different sides of the same organ, is what that is that you're seeing. Okay, but that is a good question. That's a simple cuboidal on the other side. Go ahead.
Where do you find simple squamous?
I'm about to tell you where you find all four of the simple tissues. So let's go to that next. Okay, so simple tissues. Simple tissues have one single layer. So what do you think they are going to be good at? I'll just give you two choices, and of course there's more than one thing that they're good at. But if you have a simple tissue, do you think it would be better at protection or transport? Transport, exactly. They're not very protective tissues. You are going to find them in locations in which you really want to move things quickly from one side to the other of the tissue. Okay, so let's look at a simple squamous epithelium. So here's simple squamous epithelium in one figure. Okay, that tissue you are going to find lining the alveoli of the lungs, where you do gas exchange quickly in and out, okay? You'll also find it forming the capillaries. Capillaries need to do gas and nutrient and waste exchange really quickly. So let me show you another view of simple squamous epithelium. Now this is a little hard because you're not used to looking at it in this type of section, but that is simple squamous epithelium. That's a what section of a tube? Cross section. That's actually a capillary, by the way. So you'll end up seeing that all the time with capillaries. So you're going to find it in locations where you need to do very, very quick gas and nutrient and waste exchange. and you're really, really not worried about abrasion or protection or anything like that. Okay, so that's a simple squamous epithelium. Now, some people like to ask simple squamous epithelium instead of from a side view, they ask it from a top view, and I will never ask you from this view. Although, of course, you can see a lot more of it. But my conceptual problem with that is if I'm looking at it from a top view, I can't really tell that it's either simple or squamous from a top view. So really all you're doing is memorizing a picture. There's not actually any thought or understanding that goes into that. Now, your lab instructor may ask you from that view. I don't know, but I certainly won't ask you from that view. It is a simple squamous from a top view. Okay, so that's simple squamous. Simple cuboidal is the one that's next. Simple cuboidal epithelium is right here. I just think it's the cutest tissue. It's just the cutest one. The cells are usually nice and round and they're perfectly symmetrical and they just look kind of happy. Okay, so this is simple cuboidal epithelium. So you can tell it's simple, right? You would guess that it's cuboidal. And the next question is, what type of section of this tube is this? Long section, a longitudinal section. But I can also do a cross section of the same kind of tube and still ask you. It would still be fair game. This is also simple cuboidal epithelium. Do you see it? Yeah, that's simple cuboidal epithelium as well. And so is this longitudinal section. So I always just like in my head find the free surface, count the layers, look at the shape, acknowledge that I know that this is yet another example of simple cuboidal epithelium. Is that okay? Now this website also has this picture, but it's just rude. I would never ask you from that picture. Can't see a damn thing in there. Okay? So you find this tissue, simple cuboidal epithelium, lining kidney tubules, and you may not know anything about kidney tubules, but there's microscopic tubes in your kidneys where you do exchange to get stuff out of your bloodstream into your urine, and that's where it happens, right? Like that's where you would get rid of waste products and excesses at your kidneys, and they're called kidney tubules, and there's just miles of them. There's tons of them. So that's simple cuboidal epithelium, and now let's look at simple columnar epithelium. With simple columnar epithelium, there's actually two different kinds of simple columnar epithelium. So let's look at the textbook figures first. So there's two different kinds. So by looking at this, would you guess that this is simple columnar epithelium? Yeah? Okay. Now this one's going to be different, but still, is it simple? Yeah? Does it look to be more columnar than anything else? It is. It's a different category of simple columnar epithelium, so now is the time that I teach you the difference between microvilli and cilia. So there's two different types of simple columnar epithelium. One of them is called ciliated simple columnar epithelium, and the other one's just referred to as simple columnar epithelium. So here's what we have. We've got two different tissues, each of which has columnar cells. Relatively tall and skinny, usually oval nuclei, although not always oval nuclei. This one, however, the superficial surface of each cell is highly folded. And with this one, the superficial surface is not folded, but it does have little hairy projections that look like they're coming off the top. Okay, so here's A and here's B. There's two different types of simple columnar epithelium. One of these, A or B, has a greater surface area and therefore would be much better at transport. Which one? B, exactly. Does that make sense? So this type has what we call microvilli. Microvilli are not about wiggling things around, they're just about increasing surface area, so have more room for transport. This one, does it surprise you, is actually found lining the small intestine. That's where you do most of your absorption of nutrients, right? Okay, so it increases the surface area so that I get better absorption, okay? So microvilli is to increase surface area. This one is not about surface area at all because these guys are not actually extensions of the cell membrane. They're little proteins sticking off from the cell membrane. They have mitochondria associated with the bottom so that they can wiggle. So these are about moving things around outside the cell, Okay, those are called cilia. And now I'll tell you where you find this tissue. This tissue, among other places, is found lining the fallopian tubes or oviducts. Can anybody tell me why you would want this tissue lining the fallopian tubes or oviducts? Yeah, because it's actually going to move the egg through the fallopian tube or the oviducts. That makes sense? So they have two different functions, two different structures. This one kind of looks like Bart Simpson, right? And this one's like more like a minion. Okay, but how do you tell the difference when you're actually looking at a histology slide? So this tissue right here, lining the small intestine, has, do you see that it doesn't actually look like messy on the surface, but it does look kind of like somebody took a highlighter and highlighted the surface? Okay, this one looks like this tissue has bedhead, right? It's like all messy looking, right? Okay, so you can tell the difference between the two of them as long as you give yourself some practice and look at more than one slide. Okay, so go ahead.
That other one was ciliated, right?
Yeah, this is ciliated simple columnar epithelium found, for instance, lining the oviducts. This is simple columnar epithelium. They don't say microvilli, they just assume that this is the one. This one's found lining the small intestine. So let me show you what the microvilli look like if you look at them more closely so you can get an appreciation of what's actually going on there. If you use a different type of microscopy instead of light microscopy, you use transmission electron microscopy and then blow it up. This is what the microvilli actually look like. They're little folds of the cell membrane. Tons and tons of little folds of the cell membrane, but they're very, very regular, so they don't look like messy. Okay, that makes sense? So that's cilia versus microvilli. So there are two different tissues. Now, notice, please, that I have been telling you the locations that these tissues exist. You are going to have to know specific locations at which tissues exist. And you have to be specific about it. So what I want you to imagine is when you tell me where a tissue is, you imagine me playing Magic School Bus and try to get there, right? So you need to be specific. So if you said that simple squamous epithelium was found in the blood vessels, you don't get credit. What do you have to tell me so that I can play Magic School Bus and get there? Lining the capillaries, right? If I said that a ciliated simple columnar epithelium was found in the oviducts, conceptually, you should understand now that immediately below that is going to be a connective tissue, and then there's going to be smooth muscle on the wall, and then some other tissue. So do you think just saying oviduct is enough? What did you say? Lining of the oviduct, right? So you're going to speak clearly enough that I could actually find my way there with your directions. Go ahead.
What's an example? Where can you find simple cuboidal?
Simple cuboidal epithelium is found lining kidney tubules. Okay, and we'll practice that before you have your quiz. So you just have to be really specific about what you're talking about where they're going to be found. And generally speaking, unless it's only found in a few really specific places, you can memorize one and write it in the fill-in-the-blank question. But the ones that, like the examples that I give out loud, I think they're fair game because I said them out loud, right? Okay. Other than that, you can write one down that I didn't say out loud as long as it's correct. It's totally cool. I don't mind because there's more than one place that these things were found. There are a few tissues that are only found in one place in the body, though. Okay, so we will stop there and we will pick up with the stratified tissues next time. Oh, by the way, here's cilia in action, which is kind of cool. It's all spazzy. No class on Monday. It's Labor Day. So I'll see you guys Wednesday. Have a good long weekend.
Do we have a quiz every day?
No. You have quizzes every week, week and a half. I always tell you before it's coming. Usually I try to have a quiz when I feel like the material is getting deep enough that I really need you to go study it and digest it, and then we'll have a quiz.
Our final, is it on Monday or Wednesday?